The purpose of this podcast is to educate and to inform. This is not a substitute for professional medical care and is not intended for use in the diagnosis or treatment of individual conditions. Guests on this podcast express their own opinions, experience, and conclusions. The mention of any product, service, organization, activity, or therapy should not be construed as an ASCO endorsement.
Welcome to JCO's Cancer Stories, The Art of Oncology, brought to you by the ASCO Podcast Network, a collection of 9 programs covering a range of educational and scientific content and offering enriching insight into the world of cancer care. You can find all of the shows, including this one, at podcast.asco.org. Today, my guest on this podcast is Dr. Bruce Chabner. Dr. Chabner is widely considered frankly, if not the father of our understanding of the pharmacologic principles of anti-neoplastic drugs, and probably more importantly, translation of these principles to the intelligent application of the agents we use in clinic every day. Among the many accomplishments that Dr. Chabner has had, working with his mentor, Dr. Joseph Bertino at Yale, he developed the concept of high-dose methotrexate glucovorin rescue that was completely based on their preclinical understandings of the mechanism of action and, more importantly, resistance to this agent. Dr. Chabner was also instrumental in the development of paclitaxel when he was at the National Cancer Institute, and he was intimately involved in many of the early studies that led to better understanding of AIDS and the ways to treat it. Dr. Chabner was raised in Shelbyville, Illinois, and I'm going to digress for a moment because I've always had a very special soft spot in my own heart for Bruce Chabner. A, because of his science, but more importantly, because I was raised in Shelbyville, Indiana. Both of these towns were named after Colonel Isaac Shelby, who was a hero in both the Revolutionary War and the War of 1812. By the way, Dr. Chabner, I know you were a big student of history when you were in college, so I thought you'd like this. Dr. Shelby became the first and then the 5th governor of Kentucky, and as a citizen, he was a land surveyor. There were actually 9 counties and 11 cities and towns spread around the eastern and Midwest eastern and Midwest regions that are named after Colonel Shelby. And I don't know about you, Bruce, but I had this drilled into my brain in 8th grade history class. I had to learn all about Colonel Shelby.
God.
Anyway, so he and I are brothers in Shelbyville. Dr. Chabner received his undergraduate degree at Yale, where, again, he spent a lot of his time in the history department, but also in the biology department. And then he got his medical degree at Harvard, where he stayed to complete his residency in internal medicine at the Peter Bent Brigham Hospital before it became the Brigham and Women's Hospital. In 1967, Dr. Chabner became a clinical associate in medical oncology at the National Cancer Institute, where he was, in succession, a senior investigator in the Laboratory of Clinical Pharmacology, chief of the clinical branch of the Clinical Oncology Program, Associate Director of the Clinical Oncology Program, and then he succeeded Dr. Vince DeVita, someone I have previously interviewed for this series, as Director of the Division of Cancer Therapy. In 1995, after 25 years at the NCI, he moved to Boston as the Chief of the Division of Hematology Oncology and the Clinical Director of the Massachusetts General Hospital Center, where he is now the Clinical Director Emeritus. Dr. Chabner has authored, I counted, over 500 peer-reviewed papers, I think even more than that. By the way, his first was in 1969, a case report of shaking chills related to occult lymphoma, authored with Drs. DeVita and Joe Fermini of the Lee-Fraumeni syndrome. Bruce, that really shows how old you are. He's been the editor of all 5 editions of the Principles and Practice of Cancer Chemotherapy and Biological Response Modifiers, which I consider the Bible of cancer pharmacology. And I'm looking at my 5th edition on my bookshelf right now. He's trained too many fellows for me to name, but numerous of them have gone on to be cancer center directors, chiefs of division, department chairs, and other leaders of oncology in the world. He's won way too many awards for me to go through, but he received a Karnofsky Award from ASCO, and he served on the ASCO Board of Directors. Dr. Chabner, welcome to our program.
Thanks. That's a lot of history. Speaks to my age.
Well, that's the problem of interviewing all of you folks. It takes a long time to get through all the things you've done. It's a good problem to have, though.
Yeah.
First, I want to start out. I understand you carry the flag rank of Rear Admiral. And
Right.
I want to know, have you ever even been on a ship? And more importantly, did you and Dr. Shelby actually serve together in the Revolutionary War? I couldn't figure that out.
I never bumped into him when I was on the battleship Shelbyville, but who knows? He seemed to be what I call a name dropper. He left his name on so many different things. And I think there's a Shelby County, Tennessee, which is Memphis. Plus, I think the smallest thing that he ever created was Shelbyville, Illinois, which was even smaller than your hometown.
Yep, that's true. All right, that's the last joke I'm going to tell in this interview, but I like that connection. Anyway, how did a guy from Shelbyville, Illinois get to Yale and then Harvard and the NCI? And more importantly, what made you decide to be an oncologist? I know your father was a general practitioner, but at that time, the field barely existed. What was your motivation?
Well, okay, I'll tell a bit of a story. My mother came from Chicago, and she had a brother who was pretty smart, and he went to Harvard. And he used to come down to Shelbyville because he liked the pies that she made. We were 200 miles south, so it was a trip for a pie. But at any rate, we used to play chess together. And when he was a college student and I was like in fourth grade, I beat him in chess. And he said, "Jesus, you ought to go to an Ivy League school." So that put the idea in my mind. And then my parents were not really happy with that. They wanted me to go to Washington University or University of Illinois, but I wanted to get away from home. It was a little bit confining to be around my parents for the rest of my life. So I applied to Yale, Harvard, and Princeton. And the deal was I could go to a school if I got a scholarship. So I got a scholarship to Yale. So I went there. I was happy with that choice. It was sort of, life-changing, actually. Stayed on the East Coast, and but I still have many good friends from my Shelbyville days. We all get together once a year to play golf and poker and tell life.
Yeah. So I have to interject. My father told me I could go any college in the United States as long as the tuition was the same as in-state tuition at Indiana University, which at the time was $400 a year.
Yes.
So I ended up going to Indiana University. So how did you get into oncology?
So, when I was at the Brigham, I got interested in cancer, although there was not much going on there. But one of my residents was a guy named Jack Moxley, who had been a part of the initial study with DeVita and others, George Canellis, of the MOP treatment for Hodgkin's disease. And I got really interested in that. And actually, during my internship, my sister got a mediastinal tumor during her pregnancy, and it turned out to be a thymoma. But cancer really intrigued me at that point. And we all had to apply for positions at NIH as a way to get out of the draft. And I wanted to do research, so that really appealed to me. And I actually applied for cardiology and cancer. And I was interviewed by Gene Brownlow for cardiology, and he didn't seem very impressed. But the cancer people did like me. So particularly George and Vince, who come back there, and they were young and energetic, and they had, interesting ideas about combination therapy. So I ended up in oncology.
Yeah, well, I've talked with some of the other people I've interviewed about the so-called era of the yellow berets, and how that it really transformed American medicine, in my opinion, because so many smart people went to the NIH to stay out of Vietnam. It's probably the only good thing that came out of the Vietnamese War, as far as I can see, and especially the NCI. So when you went to the NCI, Holland and Freyreich, I believe, were gone. Yeah. So you've already started to set. It sounds like Dr. DeVita and Dr. Canellis were the movers and shakers at the time. Is that fair?
Well, they were really young. I mean, it was like working for, contemporaries. There were no old people there. And, Frey and Freyreich weren't that old at that time. They were in their 40s, but Vince and George were in their mid-30s. And I was 28 years old, I guess, when I went down there. I loved it. We had laboratory opportunities. We had patients. We had people that believed that they could change the way cancer was treated. George and Vince, particularly Vince, was so energetic and so committed to the idea of changing therapy, and particularly combination therapy. So, and then the other thing that made it such a great experience were the colleagues that I had in my first group of clinical fellows. Bob Young was part of it, and I became very close friends with Bob Young. And in the same group, David Livingston was my next door neighbor, and we had interned together and been in resident together. So we had just constant stimulation from a lot of different people, all of them energetic and interested in research.
Who else was in your class besides Drs. Livingston and Young then?
Phil Shine, who made a name for himself in toxicology and then in industry. And let's see. He
was director of the cancer center at Georgetown for a while.
Yeah, right. Subsequently, there was just a long list of wonderful fellows. When I came back, I actually spent 2 years at Yale between my NCI time and then coming back to NCI, and I had a wonderful time with Joe Bertino. He was, I think, very important to me because he was really a great scientist, and I learned a lot about biochemistry, enzyme purification, and working in the lab. And so when I came back to NCI, I had sort of converted to being an anti-folate person from being interested in alkylating agents. And I was always interested, I guess, in antimetabolites, but that was a great anti-folate experience with Joe, high-dose methotrexate. It was really his idea, not mine, but the thing I worked on was the clinical pharmacology and trying to figure out why it was so toxic to kidneys. So we actually did some really interesting experiments. We gave high-dose methotrexate to monkeys, and then when they died, we took their kidneys out and looked at them, and we were doing it because we thought we would see interesting pathology. What we saw were a bunch of yellow gravel in their tubules, and it turned out it was methotrexate, and it became obvious what was happening. The drug was precipitating in the acid urine environment. But that was sort of the beginning of the methotrexate studies.
And personally, I don't think we teach pharmacology very well anymore. What made you want to go to high-dose methotrexate?
Interestingly, I was particularly interested. Joe was trying head and neck cancer. There was almost simultaneously an article from Fry and Isaac Gerasi about adjuvant therapy of osteogenic sarcoma. And there were several interesting things about that. One is it turned out that 12 patients weren't all patients with osteogenic sarcoma, but prognosis of those patients wasn't apparently obvious, but there seemed to be some success with it, and there was a lot of toxicity that they didn't really know how to deal with. And so I started doing pharmacokinetic monitoring in patients that we had that were on the treatment, and found when they went into renal failure, they just didn't clear the drug. The drug was hanging around for many days, and they were getting this horrible toxicity. So we got into this business of why the renal toxicity and the need for hydration and alkalinization particularly.
And so first of all, I have to tell you, I blamed you for much of my first year as a fellow because we had to draw the bloods. There were no study coordinators. Dr. Fry had us run around at all times of day and night drawing blood on patients who are getting high-dose methotrexate. I still mumble under my breath when I hear your name.
Well, you don't have to do all of that now, but in those days we were trying to get a more complete profile, so we did. There was a woman there at the Farber that was doing similar work. I can't remember her name, but
Sue Pittman, I think, right?
Yeah, Sue Pittman. That's right. Yeah. But that was sort of the introduction to the anti-folates. And then I got into a very interesting area of polyglutamation and how it changed the potency of the drug and led to retention, and it was an important determinant of response. That was quite an interesting area of research.
Were you the first to report amplification of DHFR?
No, that happened in 1978. I was working on MTX at the time, and we had noticed that you could select highly resistant cells in culture, but then we were interested in knowing why. And Joe Bertino had described the fact that increased hydrofolate reductase activity was found in this circumstance, but the actual demonstration of amplification in mammalian cells was done by Bob Shimke when Joe was on a sabbatical with him, and they had a medical student working in the lab on that, and on that paper that was Dan Haber, actually, who came back to
who is now the cancer center director.
Yeah,
I guess he's your boss,
it was a key
right?
paper. So we had at the time when that came out, we got interested in that, and we stuck radio-labeled methotrexate into culture with some tumor cells and found these odd migrating entities that turned out to be polyglutamates. And that led to the whole issue of what were polyglutamates and how did they change the biochemistry. And that was quite interesting. And then actually at the same time, we saw a patient. It was a young man who came to NIH with non-Hodgkin's lymphoma and was treated with high-dose methotrexate. I can't remember. I think he had CNS involvement or something like this. We found evidence of gene amplification in this patient. So it was actually the first demonstration that gene amplification occurs in people on the drug. There are a lot of interesting things that were happening at that time.
It was translational medicine before it was called translational medicine.
That was the nice part of NIH. You know, the emphasis was on the labs working with the clinics and particularly with physician scientists. So, you know, we were one of the few places where our fellows were expected to work in labs in their second and third years, and they did. And we had a wonderful group of fellows that came through. The first guy that worked on polyglutamation of MTX was Rich Shilsky,
who is now the chief medical officer of ASCO. Among many other accomplishments.
Right. But many.
Actually, I'd like to change gears for a minute, because I know you had a lot to do with the development of paclitaxel, and I've always found that story interesting. That first, that was in the bark of the Japanese yew tree, which had to do with ultimate supplies, but also the first phase 1 trials, which were some of that was done at the Dana-Farber when I was there. Can you just walk through the history of paclitaxel? I think our listeners would love to hear this.
Well, it was an accident of history, believe me. The thing started in 1964 when a group at Research Triangle, a chemist, isolated this compound from the yew tree. And they didn't actually know what it was, but it was cytotoxic, and it was an antimitotic. And it took them 7 years to figure out the structure. So finally, in 1971, a guy named Wani from that group published the structure. It was a ridiculously complicated structure, and nobody could synthesize it, at least at that point. It hung around in the lab, and nobody was interested in developing it because it was such an odd molecule. It was insoluble. Nobody could put it in solution. So it really wasn't an attractive pharmaceutical. And the thing that happened was in 1978 or 9, we had a very hot drug. It was called maytansine. And we were very eager to put this into the clinic. And it was an antimitotic also, and very, very potent drug. And so Dr. DeVita asked me to personally shepherd this thing, and he told me that he didn't want it to fail. And so I put it into patients, and it was terrible. And I kept telling him, this is not going to work. And he said, it's got to work. He was pretty persistent. Well, it didn't, and he was very disappointed. So was I. And the fact is, we had nothing else to put into the clinic at that time except for Taxol. So we said, oh, well, we'll try it. And we put it in a lipid emulsion. It was like putting it in engine oil or something. But it went into the clinic in several places. Peter Wernick did it at Einstein. I guess you guys did it at the Farber. And it was causing all sorts of hypersensitivity responses. It looked impossible. And it took about, 4 or 5 years to get it into a regimen that was tolerable. And there had been responses, The first response was in melanoma, so we were all excited about that. That was the usual circumstance in those days, that when you took a drug into the clinic, melanoma would be the first response, and no one else for melanoma ever responded.
Yeah.
But then Peter began noticing responses in ovarian cancer, and a regimen was worked out with antihistamines, so that it was reasonably tolerable. And finally, in 1991, which was 8 years after it went into the clinic, we finally decided, well, it was time to license it to industry. We did it. There was no patent, but we did it under a cooperative research and development agreement. And the only company that was interested in the U.S. was Bristol Myers. Everybody else said, this is ridiculous. Nobody wants this drug. And it was too hard to make it. You had to make it from bark of plants, and it was insoluble, and it was caused hypersensitivity. So they took it. And about a month afterward, there was a report from MD Anderson saying that it was active in breast cancer. And at that point, it just took off like a rocket. And, tried in all sorts of different diseases, was active in lung and bladder and I can't remember all the other things, head and neck. Anyway, it became the first billion-dollar drug in the cancer drug industry. And I think, there are two things that really set off industry to be interested in cancer. One was the fact that you could actually make money on it. And the second was the notion of targeted therapies, which was growing at that time.
So to my knowledge, this is the only time somebody at the NCI had to work with the U.S. Forest Service and the Bureau of Land Management regarding a new drug. Can you tell that story?
Well, there was, yes, we had the only place we could get the raw material for the drug was from the U.S. Forest Service. And so the Texas plants were being sort of cut and burned because they were considered scrub and not worth anything as lumber. So they were cooperating, and finally, when we licensed it, Senator Ron Wyden, who's still in the Senate from Oregon, got interested in this whole thing. He said, "Why isn't the government making money on this license? Why did you license it to Bristol Myers and you didn't ask for anything back?" And we said, "Well, that's not the function of NIH. We didn't have a patent. I guess we could have asked for a slice of the pie, but we didn't because no one else wanted it. We really were trying to give it away." And he was giving us a really hard time at this hearing. And then the key thing that happened was a woman who was a forest ranger with ovarian cancer. We found this woman, and she testified at how much good it did for her. And that sort of stopped all the fuss about the license. And we actually, it was the first drug where, as part of the licensing agreement, we had the chance to fix the price or agree to the price that Bristol Myers fixed. And, the government never has done that since that time. Of course, this was a circumstance where we sort of owned the information. Yeah. So they had to listen to it. But they set the price at $2,000 a course. And we considered that pretty high, but it was sort of equivalent to what other drugs were costing at the time. So we let that go by. But ever since then, the price of drugs has just escalated remarkably.
I hope there are some young people listening, because this story, in my opinion, the story of trastuzumab, Herceptin, again, is, I think people think that these things just happen because the system makes them. And my experience is they happen because the drug or the treatment gets a hero, gets a champion. Ultimately, the science has to prove it works. But I'm sure lots of people wanted to walk from Taxol. And
everybody thought it was a dog because it was, caused hypersensitivity. It wasn't all that active in initial testing, and it was really hard to make the stuff. But
well, the same thing is true with platinum. That
yes.
I did my residency at UT Southwestern with Donald Sullivan as the chair of medicine. He was a renal guy.
Yeah.
I had a patient with metastatic, and I had gone to Indiana, so Einhorn had taught me how to do it. Dr. Einhorn taught me how to give it. So I had a patient come in with widespread testicular cancer. I wanted to give him cisplatin, And Dr. Selden wouldn't let me do it because it would hurt his kidneys.
Oh,
and I said, Dr. Selden, he's going to die if we don't do this. And he said, he'll die anyway. And so I did it behind Dr. Selden's back. The guy had a complete response. Fortunately, in those days, the residents didn't have a lot of oversight, so I could do what I wanted to do.
I remember.
There were a lot of people that thought these drugs should be shut down, and it took the courage that you guys had back at the NCI and other places to push them out.
Dr. Selden was very anti-cancer chemotherapy for the rest of his life. And it was odd for me because I actually had a relative that was on his faculty, and I went down there a couple times to talk, and I always felt very uncomfortable with him that we were accomplishing something. Because his concept of success in science was getting an R01 in your lab.
He finally came to terms because Shelsky, myself, Fred LeMaitre, a number of us went into oncology and have been reasonably successful, and I think he decided that it was worthwhile after all. But it wasn't easy for him. We lost him a year ago. I still miss him.
He was an amazing guy, but he didn't. He really had a hard time believing in cancer.
So the other question, I want to change gears a little bit because I know just about the time you became the director of the DCT was when the AIDS epidemic was exploding in the early 1980s. That must have been a very confusing situation about who should be in charge of this at the NIH, which institute, and how you approach it. Can you give us some background on that?
Again, it was really a crazy time because I remember one of the first patients that was identified as having AIDS was a person admitted to the immunology branch at the NCI, not the medicine branch or the clinical branches. It was a patient who had disseminated tuberculosis and it had no CD4 cells. And everybody said, "Oh my God, what is this? This is really, really a weird circumstance." And then other people began reporting this from San Francisco and New York. So we actually, DCT, the reason we got involved was because of Bob Gallo. Bob Gallo had discovered the HTLV1 virus, which was causing this lymphoma in T cells. And we suspected that this might be a syndrome caused by a T cell virus. So in 1981, really quite early, we convened, I think, the first meeting about the biology of what was called HTLV2, I think, at the time, or 3. I can't remember which one it was. But at any rate, there were a cadre of people at NIH that felt that it was caused by inhaling gases or I don't know. There were various weird theories about it, but the theory that made sense to us was that it was caused by a virus. So Sam Broder was collecting samples from patients and brought them over to Gallo's lab. And, of course, Gallo mixed those samples with the French sample and found virus and then made a test kit for the virus, which was really a key event in beginning to control the epidemic. And because of all that work going on at NCI at the time, we were asked, and we had the only drug development system at NIH. We were asked look, can you set up a drug development system for this? And Sam Broder set up assays in T cells, infected T cells, and showed that certain nucleoside analogs could stop the virus from replicating. The first one was AZT, and his first study was in, I think, was 16 patients with AIDS, in which he showed that the T cell counts recovered and people didn't die. And from that point on, we were getting significant funding for doing research on treatment development. And it was done in conjunction with the NIAID and Tony Fauci. One of the first, what Tony did is he delegated a fellow to work with us and sort of be the liaison. And the first fellow that did that was Margaret Hamburger, who became the director of FDA subsequently. And subsequently, 4 other people from our division actually one from MGH, became directors of FDA. Ned Sharpless most recently, and then Steve Hahn, who was a fellow in the medical oncology group at NCI.
Yeah, he's just been named. It was
an unusual breeding ground for people interested in therapeutics.
That's interesting. I was a third-year resident at UT Southwestern. I was at the VA in March, and a young man was admitted to our service. He'd been a Vietnam veteran, and he had red splotches all over him. So I called a dermatologist who biopsied it, and I got a page from the pathologist, and I called him back, and he said, "You've mislabeled the samples." And I said, "What do you mean?" He said, "Well, this says it's a 37-year-old man.
Yeah.
This is something I've never seen before, but I looked it up. It's called Kaposi sarcoma. And that only happens in old men or people from Africa."
Right.
And I said, "I don't think we mislabeled things." And I think he was probably the first man in Dallas to be diagnosed with this, because it was just as the MMWR and then the New England Journal paper came out a few months after that. So, I again, for the young folks listening to this, and you've already hit this a couple of times, it's one or two patients that pique your interest that often change the world in terms of, I wonder why that happened. And
absolutely. I mean, a lot of this is an outcome of the fact that you have research people as physicians who are working with patients, and then they ask questions.
Yeah, one of the things I've carried forward, Dr. Fry used to always say, think like a scientist, think like a doctor, and ask yourself, so what? And I know you do that because, you've already told us today, and I've seen you do that in other places. The so what? Why did this happen to this patient?
Why did it happen? Yeah.
And what in my lab actually will change that? And you guys did that in spades, I think, 40, 50 years ago. It's pretty amazing.
Yeah. It's happening a lot. Yeah.
Go ahead.
No, it's really happening a lot now, but in terms of recognizing subsets of diseases, we used to think that non-small cell lung cancer was just one disease. And now we know it's 20 diseases. It's amazing. It's amazing that, as science progresses, you begin to understand the complexity of cancer, and then therapies become meaningful.
Yes, I agree.
Yeah, it's so nice. And so I wish we were curing people, but we are making a difference, and at least we're understanding.
I agree. Anyone who has not heard or read Bruce Johnson's ASCO presidential address should do so, because he pointed out exactly what you just said. He had a pie chart, and 10 years ago, the entire pie chart was chemotherapy for metastatic non-small cell lung cancer with little or no success. And now the pie chart is well over half the patients getting some kind of targeted or immunotherapy. It's pretty amazing.
It's so remarkable for young people that are listening to this, is that there are enormous opportunities for doing even better than we did. And so we just made a start in this whole thing.
Okay. With that inspiring message, which I'm glad you said it, we've run out of time. Actually, we've come to the end of our time. But Dr. Chabner, I want to thank you on behalf of all of us who trained after you, who've learned so much from what you've done, and more importantly, the patients who've benefited from the stuff that you've contributed to the field. It's pretty remarkable and inspiring. I don't use that word too often, but it is. So thank you, and thank you for taking time today. I hope folks listen to this and say, I'm going to go back and make a difference here.
Right. Thank you, Dan. I've enjoyed it. And it's been a wonderful time in this career.
Yeah, well, my pleasure. Until next time, thank you for listening to this JCO's Cancer Stories, The Art of Oncology podcast. If you enjoyed what you heard today, don't forget to give us a rating or review on Apple Podcasts or wherever you listen. While you're there, be sure to subscribe so you never miss an episode. JCO's Cancer Stories, The Art of Oncology podcast is just one of ASCO's many podcasts. You can find all the shows at podcast.asco.org.